Hello everybody, my name is Mariam and today I'll be talking about the process of transcription, which is part of protein synthesis. So the lecture outcomes are an introduction to protein synthesis, the definition of transcription, process of transcription and where it takes place, the three stages of transcription in both prokaryotes and eukaryotes, and the difference between eukaryotes and prokaryotes transcription. Basically, the role of the DNA is to instruct the cell to make specific polypeptides and proteins. And the huge length of the DNA molecule in a single chromosome carries the codes for a very large number of polypeptides. And within this extremely long molecule, a gene is the length of a DNA that codes for the synthesis of a polypeptide. Basically, polypeptides are very variable in size, and therefore so are genes. A very few genes are as short as around 75 to 100 nucleotides long, if I'm not wrong, and most are at least 1,000 nucleotides in length, and some are more. Of course, polypeptides are built up by different reactions, but it's mostly built up by condensation reactions of individual amino acids, and until now there are only 20 or so amino acids which are used in protein synthesis, which have been discovered, of course. And all cell proteins are built from them. Now, the unique properties of each protein which amino acids are involved in its construction and the sequence in which these amino acids are joined. The DNA code is in the form of a sequence of 4 bases: adenine, thymine, guanine, and cytosine. And this sequence tells us the order in which specific amino acids are assembled and combined together. The code lies in the sequence in one of the 2 strands of the DNA double helix. So one of the strands of the DNA double helix is where the code lies in, or the code sequence lies in. This is called the coding strand. And the other strand is the other strand is complementary to the coding strand. Nevertheless, the coding strand is always read in the same direction from 5 to 3. Case for a triplet code. Basically, the DNA contains 4 bases, which are adenine, thymine, guanine, and cytosine, and with ATGC are the alphabet of these codes. So if one base equals to 1 amino acid, it's known as a singlet code, therefore 4 amino acids can be coded. And if 2 bases equals to 1 amino acid, it's known as a doublet code, therefore 16 amino acids can be coded. And if 3 bases equals to 1 amino acid, therefore it's known as a triplet code, and 64 amino acids can be coded. And if 4 bases equals to 1 amino acid, it's known as a quadruplet code, therefore 256 amino acids can be coded. Conclusion: while the doublet code has too few combinations to code for 20 amino acid, the triplet code has more, around 44 spares, and each sequence of 3 bases in DNA is called a triplet code, and in messenger RNA it's called a codon, and in transfer RNA or tRNA it's known as an anticodon. And yet it has been found that some triplet codes are punctuation, signifying a stop or the end of a gene. And some amino acids are coded by more than 1 triplet code. So basically, transcription depends on, or the beginning of transcription depends on the mRNA codons. So mRNA codons, first of all, the number of nucleotides making up mRNA must be 3 times the number of amino acids making up the protein. And stop codons are basically UAA, UAG, and UGA, as you can see from the figure. And promoter, we have 2 things in transcription, which is promoter and terminator. And promoter is where it starts. So basically, it is a specific sequence of nucleotides in DNA where RNA polymerase attaches and initiates transcription. And it also determines which strands of DNA is used as a template. And terminator is a specific sequence of nucleotide in DNA that signals the end of transcription. All right. So basically, this occurs in the nucleus, where a complementary copy of the code is made by building of a molecule of messenger RNA. In this process, the DNA triplet codes are transcribed into codons in the mRNA, and this is called transcription. It is by the enzyme RNA polymerase. In this process, the DNA double helix first unwinds, and of course, when it unwinds, the hydrogen bonds in the DNA double helix are broken, and at this site of it is broken at the site where the genes are being transcribed, and while they are broken, there is a pool of nucleotides. Of free nucleotides nearby. And then one strand of the DNA molecule, which is known as the coding strand, is the template for transcription. This occurs by complementary base pairing. First, the enzyme RNA polymerase recognizes and binds to a start signal, and the polymerase or the RNA polymerase enzyme then matches the free nucleotides by base pairing. For example, A with U. We should know that in transcription, uracil is thymine. Okay. We should know that uracil is used instead of thymine, and cytosine is used instead of guanine, working in the 5-3 direction, of course. We should know that also hydrogen bonds then form between complementary bases, holding the nucleotides in place. Each free nucleotide is then joined by condensation reactions between the sugar and the phosphate groups of adjacent nucleotides of the RNA strand. This is brought about by the action of the enzyme RNA polymerase. Once the messenger RNA strand is formed, it leaves the nucleus through pores in the nuclear envelope, and it passes to tiny structures in the cytoplasm known as called ribosomes, and ribosomes is where the information can be read, and then it's used. DNA double strand once more reforms into a compact helix at the site of transcription. Now we're going to talk about eukaryote versus prokaryote. So basically, transcription translation here are coupled, and in prokaryotes, transcription and translation are separated in time and space, and in eukaryotes occurs, while in prokaryotes RNA processing occurs. So basically, the three stages are initiation, elongation, and termination for both eukaryotes and prokaryotes. And I suggest you watch Dr. Lemia's videos and also see her presentations for further information. Thank you for watching this video, and I hope I helped you enough.